This video made possible by the Global Innovation Exchange. Visit gixnetwork.org to learn more.
It's a great pleasure to be here. Thanks to GeekWire for organizing. Why don't you guys go ahead and come on up here. Desney Tan from Microsoft Research and Chad Robbins, the co-founder and CEO of Adaptive Biotechnologies. The first thing I'd say is that we're not quite talking about the tricorder here. I think that's still a long way off. Andrew, in his opening remarks, alluded to some of the barriers in our healthcare system that make it hard for data to flow the way a lot of people in tech would like it to. But there's also just a whole bunch of scientific challenges that need to be worked out. So this idea of like continuous data capture and environmental censoring and non-invasive detection of all these biological molecules to tell you what's going on in real time, long way away. But there is interesting work going on, and these guys have a very cool collaboration that they're working on around what you can call a universal diagnostic powered by AI. This is a place to start. And now I'll just start by telling you a little bit about the vision. They can tell you more about how we get there. But I wrote about their partnership back in January when it was announced. And it said, my story was that one of the dreams of immunology goes something like this. You walk into a primary care physician's office to get a universal immune checkup. They take blood, and a genome analysis looks at your adaptive immune system, the T and B cell receptors and antibodies that we make that are always adjusting to the environment around us. And the information in that blood could theoretically tell us a litany of things, like whether you had chickenpox as a kid, or whether you need it's time for a vaccine booster, or maybe you've got some emerging autoimmune disease like Crohn's disease, or maybe there's a touch of prostate cancer lurking there that you ought to keep an eye on, or maybe even treat. So this is personalized medicine on steroids. Adaptive has the chemistry and biology experience going back 10 years. It's a spin-out from the Fred Hutch in gathering some of this information from the blood or bone marrow biopsies or different kinds of samples. And Microsoft, we know, has a lot of cloud computing infrastructure and power, and a lot of smart people who seek to analyze cool data sets and apply artificial intelligence to it. So Maybe we can just start. Maybe Desney, you want to start on why did I know Microsoft has had interest in biology going back to when the Human Genome Project was just a twinkle in a lot of people's eyes, including Bill's. But it's been frustrating. It's been hard to find a really good project to sink your teeth into and make headway. So what attracted you and your team to this particular challenge at this time?
Yeah, thanks, Luke. Actually, before I say anything more, kudos to GeekWire for wonderful summit. Thanks to Regents for sponsoring this session, and thanks to you guys for showing up. So to touch on the tricorder, and I won't linger on this too much because the analogy actually breaks down in many ways, but the piece that we really enjoy about the tricorder is that the three pieces are actually sensing, storage, and compute. And therein lies the basis for this partnership, where Adaptive is coming in with a pretty novel set of sensors, the ability to read our immune system. Microsoft is bringing the storage and compute, the ability to store large chunks of data and to process it with AI or machine learning in many cases. The appeal here is pretty straightforward. As we see healthcare progress, we see a shift from otherwise fairly subjective analysis by human beings typically to what we would refer to as data science. The aggregation of large amounts of data from extremely new sensors that give us extremely new views into the world, and the ability and the assets that we have to really process these things and derive insight from them. Maybe Chad can actually say a little bit about, it'd be worth sort of framing the high-level challenges and vision a little bit more deeply as well.
Yep. Yeah, sure. Well, first of all, it's important to understand the scale of the project. And Luke went right to, okay, once you have it, this is what you do with it. And what you do with it is coming up with a universal diagnostic for disease, which I think was articulated really well. But before we get there, that blood sample and analyzing that DNA has to reference something. So that's the first step, is really building what we call this immunomap. So just to put in historical context, the human genome was sequenced around 2001. It cost $3.5 billion, but it actually missed your immune system, because your immune receptors, which are responsible for finding, killing, and then remembering your foreign bodies, or what we call pathogens, that was actually missed because those receptors, actually the DNA rearranges. And so the first step really is to be able to map that. And we're trying to map that disease by disease. And so if you think about the problem space, there are 10 to the 15th receptors, just a trillion receptors that bind to about a million or so pathogens. So what that means is that you have many of these receptors. Think of these kind of like as a scanner, if you will, or sensor, as Desney said, that are responsible for finding and locating that foreign disease-associated kind of pathogen. So Adaptive has the biology, the chemistry, and the informatics to be able to understand the relationship at scale or throughput of these receptors to the pathogens they bind to. However, there are many what we call de novo receptors, trillions of them, that you're going to see, and you don't know what they bind to. And this is where kind of the machine learning aspect come in, to be able to predict with high levels of confidence that if you see an immune receptor, you know that it's going to bind to that particular disease-specific pathogen, and therefore we're going to be able to, in some sense, reverse diagnose a disease from that. And so that is exactly where kind of Microsoft comes in, is we're feeding massive amounts of data that we can generate with our chemistry, and then Microsoft can come in and feed that into their machine learning algorithms in the black box to be able to start kind of filling in the rest of that map that we can then leverage.
Now, I should probably take a step way back here and remind people that when you look at, say, autoimmune diseases, where your immune system's going haywire and attacking healthy tissue, oftentimes that is diagnosed currently through a clinical exam. So rheumatoid arthritis, you go in and the doctor touches your joints. Like, how much does that knee hurt? How much does that knee hurt? That elbow? How about that shoulder? And they add it all up, and they try to make it quantifiable as they can, but it is not a molecular diagnostic with any degree of precision. There's a lot of subjectivity.
I'll just a quick comment on that. That's exactly right. Rheumatoid or MS. This is done by kind of rule-out tests, right? You do a bunch of other stuff, and then you settle on a disease diagnosis. Think of what we're doing as a rule-in test based on molecular knowledge and information.
And I'd be remiss if I forgot to mention that this is also applied toward cancer. The immune system is integrally interwoven with so many diseases. In fact, Adaptive just got FDA approval last week for its test for minimal residual disease. Now that's exactly what it sounds like. The question doctors often have after they treat people with cancer is, have we gotten all of the cancer with that initial surgery or that initial whack of chemotherapy? And they don't really know. You can do an X-ray or an MRI and you get some degree of specificity, but they can look at these T and B cell receptors in I think 100 or 1,000 times higher degree of sensitivity to detect those very trace cells that are lurking, that didn't quite get hit. And then the doctor can use that information to watch it or to deliver an extra round of treatment. So, I mean, this is where things are going. Much higher degree of specificity and personalization.
Yeah, this is pretty cool stuff. I mean, as Luke points out, we sort of look, we diagnose disease based on observable symptoms today. And really, we were looking for a set of sensors, and these guys come along and they say, hey, the human body is the ultimate sensor. Your immune system knows as soon as something that's not supposed to be there is in your body, it responds, and in fact, it responds in extremely predictable ways, and we can read it. That's an amazing capability to have, and it sort of transformed the way we thought about diagnostics and therapeutics and healthcare as a whole. And we're super excited to be a part of the journey in crafting not only new diagnostics, but potentially a new universal diagnostic, something that will allow us to diagnose just about all disease, infectious, autoimmune cancers, with a very simple blood test.
Okay, so you got this big, bold vision, but you have to start somewhere. It's a 7-year collaboration. There's like 9-figure dollar amount on the line here. A lot of investment on both sides. How do you get? I think Chad, your brother Harlan, the co-founder, said he wanted to make the point that this partnership is not about just going to work on something awesome. We need to get something done. There are real deliverables here. So what are those deliverables, and in some kind of sequence, if you can tell us that?
Sure. Well, the first step, and what we spent a lot of time working on this year, is just setting up the environment, really setting up the infrastructure, setting up the tools, setting up the ability to transfer data at scale. Additionally, Adaptive is in a major lab build-out to be able to prosecute the tens of thousands of samples that will feed into the environment, into the data learning and machine learning environment. That being said, we are starting to generate data in various disease states, and although it should also be pointed out that while the long-term goal is the moonshot is this universal diagnostic for disease, one of the things that I think attracted us to Microsoft, and I think vice versa, is we really wanted to be able to ship product. We wanted to have a tangible diagnostic or set of diagnostics in very specific disease states before we got to universality. And just a quick comment, if you look at kind of the FDA pathway of the MRI or the X-ray, these were actually for a specific use case. But as we all know, it's a screen of a biological system. So if you can diagnose a broken arm through an X-ray, it can be the same for a broken leg. We all know that, but that's actually not how it started. So we're going to start layering on specific disease-related diagnostics, and we've chosen certain areas to do that, and we're generating great data. And the proof of concept data that we originally generated to be able to prove to Microsoft that this was a tractable problem in CMV, which it's not that interesting to diagnose CMV because half the people have it, half they don't, and you're not going to die from it.
Cytomegalovirus.
Yeah, it's just not that big. But it was a nice proof of concept to be able to do that. And now we've taken that proof of concept and are starting to generate data sets in type 1 diabetes, celiac disease, ovarian cancer, et cetera. So these are ones we're starting with. And there's many more that we're also looking at partnerships with pharmaceutical companies, and that'll drive some of that direction as well.
Now, Desney, I think you kind of alluded to this a little bit earlier, but data analysis and AI are only going to be as good as the data that goes in there in the first place. If stuff's missing or it's inaccurate or sloppy, I mean, you're going to bake in some really bad things.
Absolutely.
So what is it? What are they doing that attracted you guys?
Well, sort of extending what Chad was saying, I think actually before setting up infrastructure, one of the things that both teams invested in was setting up culture. And it was extremely important to us that we would be working on this thing hand in hand and not with a firewall of a bridge or a lake in between us. That was the first step before even setting up infrastructure, because what we realized was that the technology would have to drive the wet labs, and the wet labs would have to drive the technology and the science and so forth. And so we really integrated ourselves as a single team. We've got offices in each other's facilities. We spend a whole bunch of time moving back and forth. The teams function about as tightly as any team can, even though we live in two separate organizations. The machine learning problem is actually pretty well posed. That was one of the things that was appealing to us. So we've got, at the highest level, what we have, and as we talk about the TCR antigen map, we've got a massive matrix. On one axis, you've got T cell receptors, and on the other, you've got antigens. And our job as technologists is to fill in this grid. right, to map every given TCR that we could see in the bloodstream to its cognate antigen. Turns out we don't really have enough throughput to do this exhaustively, and so what we get to do is actively label parts of this matrix. And therein lies the collaboration, where we go in and we say, look, if you can label this part, we could infer the semantics here, and we could start to grow the map out without all the data. And they go, well, the science tells us that we should be doing this, and we go back and forth, and we sort of develop the algorithms and the models, as Chad points out, in two different classes. One to predict disease, and that will be our initial step, taking the T-cell repertoire and predicting point diseases out of that. Eventually learning this entire map and being able to then predict the antigens, which allows us to predict all diseases.
But the number of possibilities are pretty much infinite in terms of T-cell and antigen binding combinations. So we all have a certain number of genes. Let's call it 25,000. Each of us has 25,000 genes. But those genes do not absolutely dictate what those T-cell receptors are that we're going to make, because the combinations of those genes are going to reshuffle and form something unique to address, that bacterium or that fungus that, I may have encountered in Nepal, say, that you guys didn't. I may have some different antibodies floating around in here, right?
That's why I didn't shake your hand.
But I bring that up. It's just a funny example because everybody's different. We have different code to begin with, and we encounter different things, and our immune system has this unbelievable capacity to adapt to just about everything. But that also makes it an incredibly hard math problem to figure out with any specificity. So is there kind of like training wheels version that you can come up with?
Yes, so it's not exactly infinite. There are finite combinations. It's large. So if you look at the rows and columns, by our estimate, we're talking about 10 to the 16 squared or thereabouts. Good news is there is structure in this thing. And so we do have high hopes, and we have early proof demonstrating that some parts of this is generalizable.
So one of the things that's unique about the data generation capabilities here is that we can actually do this from healthy people initially. So you can take, we're taking about 1,000 healthy individuals and leukapheresing them, basically meaning we're draining their blood. And then you can expand that blood into what we call these massive pools of naive immune receptors. So you can then use those immune receptors, and you can essentially, for lack of a better word, you can buy disease-associated antigens and run those kind of massive pools of receptors against those antigens that you buy, and then you can start really generating tons and tons of data. So you're talking about 50 million T-cell receptors per person crossed by you can go disease by disease by these pools and start generating this data. But on top of that, then you have to go and get real data sets, temporal data sets that for each specific disease, and then kind of cross-reference and check to start filling in the map further.
Okay, so you're not rate limited by samples. Sequencing, we all know, is getting faster, cheaper, better. I mean, I just saw yesterday the Broad Institute out of Cambridge says they can sequence the equivalent of a human genome every 8 minutes. It's incredible. So what's the trick? What's the really hard part that you're beating your head up against the wall that you have to get through?
Well, I think there's a couple hard parts. One is where to start. So we think that most diseases are immune mediated, but I think the signal's going to be much stronger in certain diseases than other diseases. So that's where you kind of want to start. And secondly, there are fraught political considerations, practical considerations from regulatory bodies, from reimbursement bodies, et cetera, that you want to make sure you're kind of going down the right path and kind of building this in the right way. So, for example, let's see, in ovarian cancer, we're not saying, hey, we're going to come up with early detection and initially, or a screen. We're going to say, hey, at disease diagnosis, can we move the diagnosis of ovarian cancer from stage 4 to stage 2, and where you can intervene with a treatment decision that impacts the outcome of that patient. And so we can enrich for a population and look at kind of BRCA-positive women, over a certain age, enrich for that population, and then come up with a disease. I'll say disease diagnosis in context of stage, so that we can intervene with a treatment decision that you don't have to deal with a lot of the political considerations of saying, do we have an early detection? What does that mean? Who gets tested when? Et cetera. Over time, we will move to early detection, disease interception.
Part of the issue with early detection has been a lot of false positives, false alarms, which lead to overtreatment, anxiety, more healthcare cost. A lot of people don't like that. They're going to push back very hard.
So yeah, I think that the challenges in my mind fall into 3 buckets. There's the science, there's the technology, and then there's the practice, and they sort of all interleave back and forth into each other. From a pure technology perspective, this is all new. We're sort of blazing new ground here, and at least as we've posed it, the appeal again of this is that Adaptive can generate gobs of both unlabeled data but also labeled data in a way that allows us to direct our explorations. Technology-wise, we've posed this in many ways, and it's all a big exploration right now. You could pose it as a big regression problem, right? So given a T-cell receptor, a peptide, and MHC, what is the binding energy that will pull those things together? You could couch it as a search problem. Given a T-cell receptor, what are the set of peptides that would bind to it? Or you could pose it as a classification problem, right? Given a T-cell receptor, a set of T-cell receptors, what is the disease we're looking at, or most likely looking at? And so, as Chad pointed out, we've set up platform and we've set up a huge exploration space for ourselves to go play in this. We think we have line of sight, and we think, the way we've set up the moonshot, we think we've got logical milestones and steps along the way. But there are a bunch of open problems along the way, and so that is part of the challenge.
Now, maybe this isn't a question for you specifically, Desney, but kind of what's the business case here for Microsoft? Because I see it clearly for Adaptive. I mean, you guys would be selling diagnostic tests, but for Microsoft?
It's a great question. So when our current CEO, Satya Nadella, came in a couple of years ago, he saw it as his mission to take the technology assets of a very large company, and do 2 things. One, primarily, which was sort of interesting for an incoming CEO, he said, hey, we've got to go do good in the world. And 2, in doing so, we have to create growth opportunities and new businesses that we can spawn. It so happens in healthcare the 2 align really well. And so that sort of really opens up the doors for us to commit and to do a bunch of stuff. The other piece, if you look at, maybe by example, this is sort of the best way to describe this, but probably our most recent example of this was a partnership with a company called Altera. This was in the silicon and core compute infrastructure space. Altera was making reconfigurable hardware, and we had for years identified that Moore's law, the doubling of compute every 12 or 18 months, was going to come to a natural end. Physics was going to bound us. So we'd been exploring a bunch of new technologies, and what we decided very early on, instead of trying to create this in-house and compete with some of our best partners and customers, we would partner with companies that were already on path to doing great things, and we would power and accelerate their ability to do this. Fast forward a couple of years, Altera ends up getting acquired by Intel by nearly $17 billion, one of Intel's biggest acquisitions. Altera wins. They get acquired, lots of money and the ability to scale. Intel wins because they got a new line of business. And Microsoft wins because now we can scale our ideas and our innovation through a much larger company.
Okay, now that I can see a use case there coming out of Adaptive, like maybe, if you're successful with storing all this data and analyzing it effectively, other companies like Adaptive and different applications could look at that and say, hey, we'd like to store our data on Azure and pay whatever you got to pay to do that, and also lean on your AI. So there is, that's part of the business.
I think I have a simple explanation. You can cut me off if I'm talking too much about Microsoft's intention here, but if you think about it, the idea is you go to a doctor's office, you get your blood sequenced, right, and you have all this information. That information then is going to reference this massive map that we built. So in kind of cartoon language, you take the blood and go, right, finds a bunch of receptors, right? And so that's going to go to the cloud. There's going to be cloud compute associated, or what they call transactions on the cloud, that will happen for every single one. And that's not insignificant. So when we built the use case or the business case that Microsoft was looking at, okay, how do we ultimately benefit from that? And kudos to the long-term vision, right? I think they're, first of all, Adaptive moved all of our existing business over to the cloud, which at this point is, a blip on Microsoft's balance sheet. But the idea is taking a tax on that healthcare economy down the road, whereby so many transactions will be, if this is successful, there'll be a lot of cloud compute transactions that happen, and that will result in actual dollars to the company.
These guys are going to change the world, and we're thrilled to be a part of it.
Why not just buy the company? Not you, Chad. Well, let's talk about the quality of the data, though. Back to something earlier. Chad, your clonoSEQ test that's now FDA approved, that takes bone marrow biopsy. So that's something from cancer patients where we want to know a specific question, like, did the initial round of treatment get all the cancer? That's a long way away from, simple blood floating through the bloodstream for average healthy people and doing a checkup. There's different kinds of information present in those different kinds of samples. So did you start here with the really most serious patients in need and as kind of that base layer before you work your way up to non-invasive early detection?
Sure. So a couple things to point out. One is Adaptive has sequenced many different types of samples for many years, including the blood. And our life cycle plan in minimal residual disease detection in blood cancers is to move from the bone marrow to the blood. But to answer your question, absolutely, yes. So we started with what again, my brother would call low-hanging fruit. So if you look at actually what MRD is, and we call this kind of in lymphoproliferative blood cancers. What that essentially means is that the cancer starts actually with an immune cell, a T or B cell. And that T or B cell winds up crowding out all your other cells. It becomes what we call a massive dominant position in your immune repertoire. And with that, when that cancer spreads, that cell carries with it your immune receptor. So it's a very simple test. It's low-hanging fruit. We're simply using our immunosequencing technology to tag that cancer at diagnosis. And then after a patient gets treated with a drug, with chemo, with radiation, et cetera, we can very accurately and very sensitively determine whether those residual cancer cells are gone. We're actually counting the cancer cells themselves. So this is a very, very straightforward application of immune sequencing technology. And then furthermore, we have a whole pipeline of diagnostics looking at kind of your immune repertoire and be able to predict whether or not you're going to respond to new classes of drugs, immunotherapy drugs called checkpoint inhibitors. And then we're also using the technology therapeutically now, too, for things like adoptive T-cell transfer and kind of the next wave of therapy, moving from CAR-T to TCR-based therapy, where you're taking, that's what Juno does, right? You take a patient's immune system out, reprogramming and putting back in. We're reprogramming it in a different way. So it's definitely an evolution of the technology, but to go to kind of this universal diagnostic required technological innovation, right? And what we're really good at is throughput, accuracy at throughput. So we can be very, very accurate at large scale. But we wouldn't have done this unless we had enough data to convince ourselves first, and then to convince Microsoft that this was what we keep calling a tractable problem. So we do think it's possible.
You know, you're using a lot of terms there that I'm guessing not everybody in the audience understands. Now, Desney, I noticed you're using terms like T-cell receptors and peptides and MHC, major histocompatibility complex, for the people who are not immunologists in the crowd. Like, I bring this up partly because there is a terminology, a language barrier, tech, biotech. This is hard stuff. And in fact, when I reported on the partnership, I sought an outside expert, Jim Heath at ISB here in town. He said, the Microsoft folks that I know are very smart, but don't know any biology at all. So this is going to be an awkward marriage, at least at first. What would you say to that? How are things going 9 months in?
I'll let him talk, but I'll just say briefly, that couldn't be further from the truth. You guys have been extremely impressive on the biology, clearly the expertise in machine learning, but the folks that are on our project are extremely knowledgeable.
Yeah, I mean, I think on average, if you take the 100 and so thousand people at Microsoft, it's true that the average biology level is pretty low. What we did was we built a team that had been around for a while. You know, Jonathan Carlson, who runs a lot of the technical part of the project, was a computational immunologist who'd been at Microsoft for a little over 10 years, working on HIV vaccines. We pulled folk from our genomics group, which has also been around for a while, hired a couple of new computational biologists. And really, again, you know, the appeal of this was that there was just enough overlap. We did not have the science or the biology that these guys obviously do, and we will never have that. But there was just enough overlap that we could get in the room together and teach each other stuff very, very quickly early on. Spent the first month doing tutorials back and forth. Right? We had the science team telling us, you know, teaching us about the wet lab process and their various assays and all the work that previously done. We had our team, you know, talking about machine learning and how we were thinking about these problems and how we really needed the science to drive the technology and vice versa. And so it's been great so far, and we continue to scale up on that front with good overlap. But you know, clearly the science and you know the clinical science happening on the adaptive side, and a lot of the technology innovation happening on ours.
Now, I know you actually have some shared space at each other's facilities. Adaptive right down here on East Lake, you guys over in Redmond. How many people are working on this jointly?
Right now, about 40 to 50, and that's going to scale. Frankly, because we're setting up the data environment, Adaptive can't yet take advantage of all the resources that Microsoft is willing and will throw into the project. So Microsoft is machine learning in Redmond, Mountain View, Cambridge, and Beijing, which they're ready, willing, and able to deploy to the project, which is pretty, I mean, they've been phenomenal partners and, you know, really awesome to work with. But so once we scale up our production facility, which will hit in January of this year, we're going to be able to tap more resources. But even currently, if you look at kind of the combined number of people, it's about 40 to 50 people working on this.
So the AI folks are over there saying, "Hey guys, give us that data when you're ready."
It's accelerating. I mean, data's coming in. We're starting to really generate lots of data, but it's going to go from lots to massive amounts next year.
Now, Chad, I asked you this earlier, but there are probably 2 or 3 other big tech companies that I could imagine you probably could have talked to and worked with. I mean, just real quickly, why Microsoft?
Sure. I mean, we actually did talk to, and we had deals on the table from other very large tech companies, and you can figure those out. Desney hit it on the head. I mean, there's a couple of reasons why Microsoft. First and foremost is the people. The people and the level of commitment from senior leadership. I mean, this, you mentioned Bill. I mean, this went up to Satya Nadella. My brother and I, presented to Bill Gates in the boardroom. We were invited in as part of kind of the healthcare team at Microsoft. So very strong level of commitment from the top levels. But they really convinced us of their expertise. It was clearly, a cut above the other teams that we've talked to in terms of actually the level of knowledge about the biology, and then the the thoughts on how to apply the machine learning to the problem, I think, was very concrete to us. And then there's the proximity, even though they have offices around the world, the proximity of actually being able to go back and forth and develop these interpersonal relationships, was a huge selling factor as well.
Well, I hope you guys commute in the middle of the day across that bridge as you're save yourself some time. With that, we have to wrap up. Thanks, everybody. Please give our panelists a round of applause.